Awesome. So I made a little box on the top right if you want to put my face there, just so it's not distracting. So I'm so happy to be presenting here on some of the research I've done while I was in Archbold earlier this year, which seemed like many years ago now, especially now that I moved up to DC and it's getting colder by the day. I really miss the warmer weather. So this is research in collaboration with Scott Egan, Amanda Driscoll, and Jim Ott. All of them are in Texas. They've been working on this particular gall system way longer than I have. I just started getting into this, but like as a taxonomist, this group really interested me. So I'll be talking about the taxonomic confusion that spanned over 160 years. This Belonocnema gall wasp associated with live oaks. Okay, so a little bit of background information. Gall wasps are in the family Cynipidae. They are one of the largest groups of gall-inducing arthropods, which means that they're capable of producing these essentially plant tumors by laying their eggs into different plant tissues, and then their larvae will be able to grow inside here, feeding on the plant tissue. So it's sort of it acts as both a source of food and housing. So think of it as like almost a gingerbread house, but for gall wasps. So Belonocnema is one of these groups that are on oaks. They are recognized by this large spur on their front leg. So they use this to dig underground. So it's really easy to recognize, even though most other cynipids are not. And they're common on live oaks in South and Southeastern US, basically around the Gulf of Mexico. And I'm sure for those of you that are Archbold, you've seen oak gall somewhere, but the ones that are made by Belonocnema, at least the ones you will see, likely see, are the ones on the leaves, these P-shaped circle galls. And like I mentioned before, my collaborators have been working on this for quite a while now. They've been using it as a model system for various aspects of ecological and evolutionary study. And this is also one of the species that Abe used for one of his burning experiments. So that was one of the species he was looking at. So there's also a genome that's being sequenced for one of the species here. So it is a really interesting model system that my collaborators are developing. So you can, it's so popular that someone even made a meme about these gall wasps. So if you like memes, I spend way too much time looking on the internet for entomology memes. So here's one for you. So Belonocnema, like many other gall wasps, have 2 generations per year. In the spring, they have a sexual generation, and you see that the gall here is a potato-like gall. And like potatoes, you have to dig underground. They're on the rootlets of oak galls, oak trees. So you probably won't be able to see these until you actually go down and start digging into the roots. But in the fall, they emerge out of the in the spring. So once they emerge out of these potato galls, they will lay eggs into the leaves, which will then form these P-shaped galls on the leaves. And then they will emerge in the fall as the asexual generation. So they only have females, where the sexual generation have both males and females. And you can see that they look quite different from each other. And this has been a problem for gall wasp taxonomy because they look different, they make different galls. So in the past, a lot of species are described under different names, or even in different genera because they look so different. And this genus is no exception. I'm not going to go into the details of the taxonomic history here. You can read it if you want to, but it's neat that this genus, the first species, is named after Mary Treat, who was a Floridian women naturalist. She first found the specimen, sent it to an Austrian, Gustav Meyer, who described this genus Belonocnema. And since then, multiple species have been described, moved around in typical taxonomic fashion. So me trying to dig through this literature, it gets really confusing, but I think I got it. So as of now, before this current study, we have one called B. falsoria, known from Florida, and only from the leaf galls. And then another one called B. treaty. It goes from Texas all the way to Florida, and it's known from both leaf and root galls. Okay, so remember that there's currently there are 2 known species. And this is corroborated by CO1 data. So this is mitochondrial DNA barcoding. My collaborators have done some work on this group, and they've sequenced specimens from Texas all the way to Florida. And surely enough, it seems like there are two species based on CO1. It matches what we currently know. But just last year, Amanda, who led this project, she sequenced a whole over 1,000 specimens from this same region from different hosts. And what they found actually is like there actually might be 3 species instead of 2. And they did this using genome-wide sequencing, which is single nucleotide polymorphism, the SNP data. So it's a lot more data. Once you throw that at it, the number of species change. So this is when I got interested. As I started rearing galls in Florida, I noticed that there are some taxonomic differences, morphological differences. So I reanalyzed their data because I just like to play with molecular data as a hobby. So CO1, I took that data, reanalyzed it. Indeed, it shows 2 species. So here you see one in orange, right? One clade in orange, and the other one in both purple and green. But even though the purple and green look morphologically distinct from each other. But if you look at the SNP data, so I reanalyzed that data set as well. I took some subsamples because 1,000 specimens are just too many. Then you have 3 distinct clades here, right? So the green and the purple separates out nicely. So this is interesting. So this is a case where you have a mitochondrial DNA and nuclear DNA discordance. And this has been recorded in other oak gall wasps. So it's an interesting case. It's caused by possibly a few factors such as incomplete lineage sorting, but we don't actually know what's going on here. But once you look at the morphology, it certainly are 3 distinct species. And it was really interesting to try to look for specimens while everything was on lockdown. Luckily, museum curators were graciously enough to either send me photos or like go in and take pictures of these specimens, including the type species that's in Austria, taken by Dominique here. So there are indeed 3 distinct species. And this one on the bottom left is the one that's mostly commonly found in Archbold. And this is the one on Quercus geminata, which is the sand live oak. And you see that they have the longest tibial spur here compared to everyone else. And they have these small twisted wings and that they actually cannot fly. So we think that both of these characteristics might be adaptation to have a better lifestyle digging underground. Remember, because they have to dig underground to lay, to induce galls under in the rootlets. So in conclusion, how many species? Well, based on our evidence here, there should be 3 in total. And you see that there are distributions here. So there's one from Texas to about the middle of Gulf of Mexico. And then you have 2 species on the eastern half that overlaps in range, but they seems to have a preference or at least or partially preference towards one particular plant host. So while doing this, we did redescriptions for all 3 of these species, both sexual and asexual generation, provided identification keys, and we corrected names in the published work because there's a lot of published work, most of them under the name B treaty. So we went through every single one of them and sorted them out in the process. And this paper should be coming out soon. Hopefully we're in the 2nd stage of revision. So future studies, while doing this work, we realized that there seems to be a hybrid zone around this part in Mississippi. So this is where 2 species, the molecular evidence seems to show that they are some mixing going on. So we want to do more sampling around there. But that'll have to wait until the pandemic's over. And with that, I just want to thank all these individuals, in particular Mark, for hosting me at Archbold, where I spent a month there collecting with my colleagues. So here's Scott and I finding our potato gall, and then Chang Ti, another Cynipa gall researcher, he came to visit me. So we have a lot of specimens that I still haven't had time to process yet, but hopefully in the next while, maybe years to come, we'll be able to get to it. Okay, thank you so much.
Thank you very much, Miles. We do have a question from Scott Ward. For insect taxonomy, does morphology and do morphometrics lead the way of names or genetic analyses flipping this understanding upside down?
Yeah, great question. So I think in the past is definitely morphology, so morphometrics, and then of course you incorporate some ecological information. But now as more genetic information comes in, you have to consider that as well, right? Ideally, you want to use all of these evidence and then you make an informed decision based on these because there probably will be some sort of disagreement. And depending on who you talk to, they might have a different opinion on which one weighs more. I definitely value morphology, but as more and more powerful molecular methods become available, I'm starting to leaning on that in addition to just morphology. And also, how deep are the potato galls in the soil? Not actually not very deep. So like, especially in the Geminata, because there's sandy soil, so you just have to brush some of the sand away to follow along the rootlets. You don't even have to like dig deep, and you should be able to find it.
Great. You saw the follow-up question as well. And I also want to point out that Warren Abrahamson has given you credit for sorting out all those cynipids.
Great.
I don't know if we have any other follow-up questions. I did have a quick question. The map of the distribution that you showed, you had, I don't remember the exact species, but you had the purple and the green overlapping. Why do you think they were able to be, or are they identified as different species, or what kind of pushed them to be separate species?
So I think this is the part where the two species are, one's on Virginiana, Quercus virginiana,
Okay.
the other one's on Quercus geminata. So there might be a host associated differentiation.